That's our PCR machine, so we can do fluorescence polymerase chain reaction. So you basically amplify either DNA or RNA, depending on what kind you're doing. And so this allows us to measure how much more we had at the start of one versus another, based on how fast or how slow it amplifies. Within these cancer cells, what they have more of and what they have less of. So we can compare that. If we give them a treatment, what's different versus if they didn't get that treatment, or comparing certain differences between different types of cancer. PCR requires probes. And so what they have is a little molecule on the probe. So when it binds to our DNA and then it gets knocked off, it releases a light signal. So it basically measures how intense the light coming off it is or how not intense it is. And how intense it is based on how much of it we have at the start versus how much we don't. This is for looking at protein levels and whatnot. So if we do a Western blot, which allows us to separate proteins based on size, it's basically like a giant photocopier, right? So we'll put it there and we'll scan it, and it'll do it similarly where you'll have a probe that binds to whatever protein we're looking at, and it has a fluorescent molecule on it. So this will excite it and it will measure if we have that and how much more we have between samples. We can put up to a whole rat in there. It can scan plates, wells, whatnot, everything. So this is a very cool machine. This is like our master mix. So everything our reaction needs, the enzyme, all the little chemicals the enzyme needs, it's all in here. And then this measures our probe. So this would be a housekeeping. This is basically similar across all conditions. Pretty much every cell expresses this. And then this is what I will be measuring. And so it'll basically use this as like a baseline value. And this will be like our 1, for example. And then compared to that, if this is higher or lower, it'll tell me if it's upregulated, downregulated, that kind of thing. So based on our PCR data, it essentially would give us something like this per well. And this is what you're starting out with, and then it's amplifying it, and then the reaction's ending. And so based on how quickly or how slowly it amplifies, which it's able to gauge based on the fluorescent signal the probes give it, it's able to tell us that you had more of this at the start and less of this at the start. And so it allows us to run certain tests in how, if we treat cells with different dosages or different rests, et cetera, like based on all what the different projects are, what differences are arising in their RNA levels. So in this one, what we did was we used this machine here. So what you're looking at is a scan of a membrane. And what each of these bands represent are different samples. So they were either control, or they were treated with a drug, or they were treated with a drug and then given some time to recover and rest from it. And then we looked at the expression of a certain protein. So as I was mentioning, one of these bands represent our housekeeping, which is this red one here. You can see it's kind of consistent all the way through. And it basically allows us to control for if we loaded more protein versus less in any of the samples. And then this was our protein of interest. So it's looking at based on the intensity. So if this is brighter than this, then the control sample had more of this protein. If it had less, et cetera, right? So it's again also quantifying it based on the fluorescence you're getting. So what we have is our protein on the membrane. We separated it based on size. And then we washed it with an antibody that's specific to that protein. So the antibody bound to it, and it has a fluorescent molecule on it, right? Which allows us to use the scanner to pick up on that, and then we can computationally analyze it as well. So it allows us to, again, so before we use that to look at RNA levels, then we can look at this to measure protein levels as well. So we're getting a much bigger and more cohesive picture. We're checking it at the RNA level, then at the protein as well.